Hey everyone, we're going to look at some basic differences between skeletal muscle models, cardiac muscle models, and smooth muscle models. Let's start with the smooth muscle. What you'll notice about smooth muscle, individual fibers are long, but they're spindly and they taper off at either side. They only have one nucleus. And the proteins, the contractile proteins inside of them, they're not quite arranged in a particular order. You just kind of form this mesh-like network. And so when this contracts, it just kind of pulls and cinches in like a mesh bag. Because these myosin and actin proteins are not as organized as in skeletal muscle or as in cardiac, you don't have striations in smooth muscle, that's why it's called smooth. Because it's not as organized, you don't get really strong contraction. But that's what we want. We don't want really strong contraction because we have smooth muscle lining areas like our digestive tract. If we want to contract our intestines here, the lining of the intestines, that would be these right here. Here is the smooth muscle deep to the epithelium, the simple columnar, and deep to the areolar connective tissue. You actually have two layers of smooth muscle. We'll get to that later. But here's smooth muscle. Here's smooth muscle. You can see how spindly it is in this section right here, and also here because this is circular. This is longitudinal. You can see how spindly these fibers are, and we want to just gently push food or waste along our digestive tract or respiratory tract or reproductive tract, places where we find smooth muscle. Cardiac muscle, what's unique about cardiac muscle, it also, like smooth muscle, only has one nucleus, and I don't see a nucleus. I guess that's probably it. Only one nucleus per cardiac fiber. Of course, where do you find cardiac muscle? You find cardiac muscle in the heart. So the walls of the heart, of the chambers of the heart, that's cardiac muscle. And the goal of the heart is to beat with a rhythm and to squeeze from a certain direction. When we squeeze the heart, we want to squeeze from the bottom up. In other words, we want these cells to go in order. This one should contract first, then this one, then this one, then this one, and so on, so that we pump blood out of these blood vessels at the base of the heart. So, what's unique about cardiac fibers to allow for that rhythmicity is that an individual cardiac muscle fiber is branched. You can see how it kind of branches off like that. By being branched, it can connect to other muscle fibers in unique ways, kind of forming a network. And when they connect, at this connecting point, which is called an intercalated disc, they connect in two special ways. One way is that they link together with proteins. That's called a desmosome. That allows them to transfer force. Think of it like a hitch. If this one contracts, it's going to pull this one with it. So a desmosome helps cells pull in unison with each other. Like if you're pulling a rope in a tug of war, if one pulls, it's going to pull you with it. But what you also find are gap junctions. And gap junctions are channels, which I'm not sure what this one's supposed to represent, a desmosome or a gap junction. But gap junctions are channels, little pores, through the plasma membranes of both muscle fibers. So that means any of the cytosol in one cell can pass through to the other cell. Why is that important? To control muscles, you need electricity. You need electrical flow. There's a flow of sodium ion going through. Ions are charged, sodium ion is a positive charge, and the movement of ions, movement of charges, that's what electricity is. So to control any muscle, you need electrical flow. If this muscle is activated, that means there's sodium flowing through, which means it can then flow through to the next one, which will help in the activation of the next one. We want to, once again, activate one cell, then the next, then the next, then the next. That's what gap junctions allow. It allows them to contract in a rhythm from one to the next, to the next, to the next. Skeletal muscle. This is skeletal muscle in cross section. You can see the cross section here, and each of the myofibrils. There we go. And here is skeletal muscle in longitudinal section. In a previous video, we talked about the coverings of muscle. Here you can see the covering of an individual muscle fiber. This is the endomysium. We actually have another model right here, cross-section, longitudinal section. You can see the endomysium covering the muscle fiber. We also know that muscle fibers are different from smooth and from cardiac because they have multiple nuclei. So here's a nucleus, here's a nucleus. The nuclei are very close and they kind of bulge out from the sarcolemma. Sarcolemma, that's just a special term for the plasma membrane. Sarcolemma is just a special term for plasma membrane of a muscle fiber. So here's our sarcolemma. To control a skeletal muscle, that requires a neuron, or any muscle that requires a neuron. When we talk about smooth muscle later, we'll see how a neuron controls this. While there are gap junctions in cardiac, we'll see how to initially activate everything. You need neuronal influence, though cardiac muscle is special because it also has autorhythmic cells that control them, and we'll get into that later. But here is a neuron's axon, and here's the axon terminal in yellow. That's releasing neurotransmitter. And in this model, we have an axon releasing neurotransmitter from the axon terminal to the motor end plate. We'll look at that in more depth in the next video.